Imagine a world without disease. Imagine rewriting the very blueprint of life itself. This is the promise and peril of CRISPR/Cas9, a revolutionary gene editing technology. CRISPR, an acronym for Clustered Regularly Interspaced Short Palindromic Repeats, allows scientists to make precise changes to DNA sequences. It is a tool of unprecedented power and precision. With CRISPR, we can target and modify specific genes, correcting genetic defects and potentially curing diseases. The implications are staggering. CRISPR holds the key to treating inherited disorders like cystic fibrosis and Huntington's disease. It could lead to the development of new therapies for cancer and HIV. The possibilities seem limitless, but so do the ethical concerns. This technology has ignited a firestorm of debate. Some hail it as a beacon of hope, a chance to alleviate human suffering on an unprecedented scale. Others fear its potential for misuse, envisioning a dystopian future of designer babies and genetic inequality. The power to edit our genes is a double-edged sword. It is a power that demands our utmost care, responsibility, and wisdom. The future of humanity may very well depend on how we choose to wield it. The discovery of CRISPR/Cas9 as a gene editing tool is a testament to the power of scientific curiosity and collaboration. In 2012, Dr. Emmanuelle Charpentier and Dr. Jennifer Doudna published their groundbreaking research revealing CRISPR's potential to revolutionize genetic engineering. Their work earned them the 2020 Nobel Prize in Chemistry, a well-deserved recognition of their profound contribution to science. Charpentier and Doudna's work built upon the earlier observations of other scientists who had identified CRISPR sequences in bacteria. These sequences, they discovered, were part of an ancient bacterial immune system, a defense mechanism against invading viruses. Charpentier and Doudna recognized the potential of this system to be repurposed for gene editing. They demonstrated that the CRISPR/Cas9 system could be programmed to target and cut specific DNA sequences. This breakthrough opened up a world of possibilities for genetic manipulation, allowing scientists to edit genes with unprecedented precision and efficiency. Their discovery was a watershed moment, marking the beginning of a new era in genetic research. The advent of CRISPR/Cas9 has not gone unnoticed by the business world. Biotechnology companies have sprung up eager to capitalize on the potential of this revolutionary technology. Millions, even billions of dollars are being poured into research and development. The race is on to develop new gene editing therapies and bring them to market. While the potential benefits of these therapies are undeniable, the commercialization of gene editing raises concerns about access and affordability. Will these life-changing treatments be available only to the wealthy, further exacerbating existing health disparities? Moreover, the pursuit of profit should not come at the expense of ethical considerations. The potential for misuse of this technology is real and must be addressed. We must ensure that gene editing is used responsibly and ethically, with the well-being of humanity as the paramount concern. To understand the profound implications of gene editing, one must first appreciate the intricate beauty of DNA replication. This fundamental process occurring within the nucleus of every cell ensures the faithful transmission of genetic information from one generation to the next. DNA, the blueprint of life, is a double helix structure resembling a twisted ladder. Each rung of this ladder is composed of two nucleotide bases paired together by hydrogen bonds. Adenine, A, always pairs with thymine, T, and cytosine, C, always pairs with guanine, G. During replication, this elegant structure unwinds, each strand serving as a template for the synthesis of a new complementary strand. An enzyme called DNA polymerase orchestrates this intricate dance, adding nucleotides one by one, ensuring that each base is paired with its complementary partner. This process, though remarkably accurate, is not immune to errors. Occasionally, a wrong base is inserted or a base is skipped altogether. These errors, known as mutations, can have profound consequences for the organism. Section 5. When genes falter, a look at common mutations. Mutations, those subtle alterations in our DNA sequence, are the driving force of evolution. They introduce genetic variation, providing the raw material upon which natural selection acts. However, not all mutations are beneficial. Some can lead to disease. Cystic fibrosis, a debilitating genetic disorder affecting the lungs and digestive system, is caused by a mutation in the CFTR gene. This mutation disrupts the flow of salt and water in and out of cells, leading to the buildup of thick, sticky mucus. Sickle cell anemia, another inherited blood disorder, results from a mutation in the HBB gene. This mutation causes red blood cells to become misshapen and break down, leading to anemia, pain, and other complications. Down syndrome, a chromosomal disorder, arises from an extra copy of chromosome 21. This extra chromosome disrupts the normal balance of genes, leading to intellectual disability, developmental delays, and distinctive physical features. Hemophilia, a bleeding disorder, is caused by mutations in genes that provide instructions for making clotting factors. These mutations impair the blood's ability to clot, leading to prolonged bleeding. These are just a few examples of the many genetic disorders that can arise from mutations. Understanding the causes and consequences of mutations is crucial for developing effective treatments and therapies. Section 6. Mapping Our Essence. The Human Genome Project's enduring impact. The completion of the Human Genome Project in 2003 marked a pivotal moment in the history of science. This ambitious international effort, launched in 1990, aimed to map out all the genes in the human genome. The project's success ushered in a new era of genomic medicine, paving the way for a deeper understanding of human health and disease. By deciphering the complete sequence of the human genome, scientists gained unprecedented insights into the genetic basis of life. They could now identify genes associated with specific diseases, develop new diagnostic tools, and design targeted therapies. The Human Genome Project also fueled the development of new technologies such as high-throughput sequencing, which dramatically accelerated the pace of genetic research. This in turn led to a surge in discoveries, revealing the genetic underpinnings of countless diseases. The legacy of the Human Genome Project is profound and far-reaching. It has transformed our understanding of ourselves and opened up new frontiers in medicine. Section 7. Playing God, the ethical tightrope of gene editing. The ability to edit our genes raises profound ethical and societal questions. Should we alter the genetic makeup of future generations? What are the potential consequences of such interventions? These are questions that demand careful consideration and open dialogue. One of the most contentious issues is the potential use of gene editing for enhancement purposes. Imagine a world where parents could select the traits of their children, enhancing their intelligence, athletic ability, or physical appearance. Such scenarios raise concerns about genetic inequality and the very definition of what it means to be human. Another ethical concern is the potential for unintended consequences. Gene editing is a complex process, and even small errors could have unforeseen and potentially harmful effects. Moreover, the long-term consequences of genetic alterations are difficult to predict. The ethical dilemmas surrounding gene editing are complex and multifaceted. We must proceed with caution, engaging in thoughtful dialogue and establishing clear ethical guidelines to ensure that this powerful technology is used responsibly. Section 8. A future reimagined. CRISPR's potential to reshape medicine. Despite the ethical concerns, the potential benefits of gene editing are undeniable. CRISPR/Cas9 and other gene editing technologies hold the promise of revolutionizing medicine, offering new hope for treating and even curing genetic diseases. Imagine a world where cystic fibrosis, sickle cell anemia, and Huntington's disease are relegated to the history books. Imagine a world where cancer is no longer a death sentence, but a manageable condition. This is the future that gene editing could help us create. Clinical trials are already underway, testing the efficacy of CRISPR-based therapies for various diseases. While it is still early days, the initial results are promising. The potential of this technology to alleviate human suffering is immense. However, we must proceed with caution, ensuring that these therapies are safe, effective, and accessible to all who need them. The future of medicine is being rewritten, and we must ensure that it is a future that benefits all of humanity. Section 9. A call to action. Navigating the uncharted waters of genetic engineering. We stand at the precipice of a new era, an era defined by our newfound ability to manipulate the very essence of life. The power of gene editing is both exhilarating and terrifying. It is a power that demands our utmost responsibility and wisdom. The ethical dilemmas surrounding gene editing are complex and multifaceted. There are no easy answers. We must engage in thoughtful dialogue, weighing the potential benefits against the risks. We must establish clear ethical guidelines and regulatory frameworks to ensure that this technology is used responsibly. The future is in our hands. Let us proceed with caution, humility, and a deep sense of responsibility for the well-being of future generations. The choices we make today will shape the genetic landscape of tomorrow.